Hi, everyone. Welcome to the Clinical and Phenotypic Data Capture Workstream. I will be doing the first part. This is Melissa Handel, and David Hansen will be doing the second part. So first, I'd like to also introduce and thank Lindsay Smith, our workstream manager, and Moni Munoz Torres, our program manager. So our work stream is really about the idea that we can go from practice-based evidence to evidence-based practice. And what I mean by that is that we want to understand the standards that are needed to take data from patient encounters, deliver them to clinical databases and other knowledge resources, such as registries, gain inference from that knowledge and create a corpus of medical knowledge that can then be managed and delivered in the form of clinical guidelines, clinical decision support, and expert systems back to the clinicians caring for the patients. And the fundamental foundation of how we think about this cycle relies upon how we develop and use and apply standards and standards such as terminologies and data models to help make this cycle possible. So this is really the work stream where we envision a number of different standards that help enable learning healthcare in the context of genomic medicine. So our work stream has one approved standard to date called Phenopackets version 1. Phenopackets is an information model that enables clinical phenotypic information to be computably and openly exchanged across systems and contexts, and I'll be talking a little bit about that. We also have under development a few other areas of work. One is a pedigree standard, and we hope that you'll join our pedigree workshops during the plenary. And the idea is to improve the way in which we represent pedigree information and exchange pedigree information in different contexts in the EHR and clinical genetics context and in diagnostic labs. With Phenopackets, we continue to extend our work. We are working on FHIR interoperability. We also are working with the ISO community to release Phenopackets as an ISO standard. We are developing enhancements to the Phenopacket schema in the areas of cancer and treatment course, which is a collaboration with the Genomic Knowledge Standards workstream. And we're also investigating the ways in which we might build a computable cohort phenotype standard, because no such standard exists across different consortia and systems for how to represent the variability, the phenotypic variability, and the inclusion and exclusion criteria for designing a cohort. So Phenopackets V1 is led by Peter Robinson and Jules Jacobson. So as we think about the number of different kinds of ways that we standardize and exchange information about genetic information, we have no shortage of them. Phenopackets really aims to do the same for phenotype data. So who are the users and what are the use cases? Well, we imagine that Phenopackets can be generated by physicians or patients and families. They can be delivered to diagnostic screening programs, used in clinical trials and patient registries. We're really excited about working with journals more recently to implement improved representation of phenotypic information in journals so that we can understand the actual phenotypic spectrum of each patient that's represented in many of the clinical genetics manuscripts that describe relationships between genotypes and phenotypes. And then we also aim to make these openly computable and available as response elements in other GA4GH standards such as and workstreams such as Beacon, but also available as outputs from any given knowledge resource, as I mentioned earlier, for completing the cycle of learning healthcare. So one example of implementation is the EBI biosamples database, where you can get a Phenopacket associated with the biosamples present in the EBI biosamples database. And also the AMED Biobank Network, which is Japan's biobank network, where for each biospecimen within the biobank network, you can click a button and get the Phenopackets clinical information. And so these are clinical computable representations of the phenotypic characteristics that relate to that biospecimen that can be used in informatics workflows, in search and discovery systems, and to identify patients that have similar characteristics around the world. And with that, I'll hand it off to David Hansen.
I'm David Hansen from Australian Genomics, and I co-lead the Clinical and Phenotype Data Capture Workstream with Melissa Handel. Ensuring that data captured by G4GH standards supports the implementation of genomics into healthcare is vital, and we want that data to be computable. That is an essential part of the work of the ClinPheno workstream. Phenopackets and its use of ontologies is obviously one example demonstrating that computability. Another of our products is to enable the standardised and computable information about pedigree and family health information to be captured and exchanged accurately. I'd like to acknowledge that great work is already underway with our project leaders, Orion and Grant, along with a really fabulous bunch of contributors. Earlier in the year, we conducted a survey across driver projects and other interested groups on their uses and challenges of pedigree and family health history information. Shown here is the key takeaway, which is a lot of groups are having challenges with pedigree standardisation. This includes hand-drawn and non-computable pedigrees, inability to represent complex families, and of course a lack of integration with analysis tools. While there is some standardisation on the PED file as a standard, researchers and clinicians report that the PED file doesn't provide everything needed and that we need to go beyond the basic parent-child relationships. Here you can see some of the types of information which are missing from even the best collected pedigree information. Orion, Grant and the team have now come up with a key set of requirements that we are aiming to support in the release of our first pedigree standard with J4GH. This includes the ability to convert a genetic family history from one proband to another, natively represent everything in the 6-column PED format, represent a set of minimal core family health history data elements, natively represent most of the information currently in the National Society for Genetic Counselors Nomenclature Guidelines, and as well work with the pedigree representation of other standards such as FHIR or Phenotypic. The good news is that this work already has driver projects involved and that we already have software that can be that implements it as part of the development of this J4GH pedigree standard. If you're capturing pedigree information in your driver project, we'd love to have you involved with the development of this, and we can provide you software, hopefully that fits with your current data collection. So what's next for the clinical and phenotype data capture workstream? At this plenary, you'll have the chance to join in a workshop we're having about computable cohort representation. And then we've already started work on expansion of the Phenotypic standard. This includes new fields to serve the cancer community to support COVID-19 research, and we want to integrate with the J4GH variant representation version 1.0 standard. We'll also continue to focus on community adoption and implementation, and really importantly, we're working with a number of other standards development organisations to take our standards through their processes. This includes ISO and HL7 with the FHIR implementation guides. So, thank you for listening. We'd love to have you involved in our work stream. We have monthly full work stream calls as well as bi-weekly project subgroup calls at the moment. That's for Phenotypic and pedigree. We have a GitHub space and we have a Slack space. So I hope to see you at one of those meetings. Thank you.